Thank you, Paul. Thank you, Paul, and thank you everybody else for coming here this evening. It's so great to see so many colleagues and the undergraduate students and the MSc students and all my PhD students here to see my inaugural lecture. And a special thanks to Paul, because I think we both started on very much the same day back in 2008, and he's been very supportive in all the time that I've been at the University of Portsmouth. So you might have guessed what I'm going to be talking about. It's the effects of pharmaceuticals like the contraceptive pill and the synthetic estrogen in the contraceptive pill and Prozac on marine organisms, and some freshwater ones as well. Normally, I would leave the acknowledgements to the end, but I'm going to go straight to the acknowledgements because I'm worried I'll get to the end and I won't sort of give everybody the justice that they deserve. So the top row, I'd just really like to thank all my past and current PhD students, and below that is the ones which I've second supervised and thoroughly enjoyed being parts of their projects as well. These are the people, and all the postdocs that have worked with me are the ones which have done all the hard work that you're going to see today. And so when I say we, it's the royal we, and it's really them that have done all the fantastic science. There's also all my great colleagues that I work with at the University of Portsmouth. I couldn't fit you all on, but there's also the biological sciences, the rest of the team there, and the school and the faculty offices, and a special mention to those in finance, because without them, I couldn't get some of the funding from all these bodies here as well, and they helped me so much in putting bids together along with the research office. Coupled with that, there's all my national and international colleagues I've worked with, plus in red here, a special mention. These 4 people were the supervisors for my PhD and gave me a shot. And this is Professor Teresa Fernandez, Professor Paul Reed, Ian Davies, and Craig Robinson when I was up in Scotland. So without them, I wouldn't be here today, so a special thank you. This is the map of my talk today. First of all, I'm going to tell you a little bit about me, probably some embarrassing photographs as well. Then we're going to talk about pollution in general, and that's where your little sachet of sugar comes in as a prop that you have for today. Don't get too excited by that, by the way. There's no coffee or anything. There is some wine afterwards. And then I'm going to talk about endocrine disrupting chemicals, or as the newspapers called them at the time, gender-bending chemicals. Prozac, and as a result of the gender-bending chemicals, I thought we may as well call these brain-bending chemicals. And then towards the end, it's National Science Week this week, and I thought I'd just put a little bit about science integrity and science communication and alternative facts, and a bit of fake news in science as well. So make it a little bit more topical. So I think the trajectory that I was on was probably from quite a young age. I've been working with crustaceans for quite some time. The bit I'm going to talk about today is somewhere between this photograph and this photograph here that was taken a couple of years ago. I was also at Plymouth University at one point, and I thought it was slightly ironic if I'm talking about sex changes, the fact that it looks like I've had one myself at one point or other. And that's my old surf club card from University of Plymouth. Now, after I finished my degree and my master's back at Swansea, I got a job as a nature conservation officer and a pollution control officer, and I had some fantastic jobs pulling out old bikes and things, cutting up burnt out cars. And if any of you visit the Gower Peninsula and you've been to Three Cliffs, there's some stepping stones just down there, and I made some of them. And so if you're ever going across them, remember that I've made a couple of those. I love the fact that I made the effort to put in some headgear here for health and safety, but was still wearing a pair of Converse with a pneumatic drill by my feet. So I finished that job, and all my friends were going around the world, and I thought, right, I want to go to a different country. So I signed up to become an expedition leader on a turtle project, which was great, and I got back, and then I had this big decision to make. There was another job in northern India, going on elephants, trying to go into the forest to track tigers. But it's a voluntary job, and I'd already done a voluntary job, and it was time to start earning some money. So I have Paul Hayward and John Lancaster thank me to try and take up a paid job at Swansea University as a marine biologist, and that is really what marine biology is like, in the rain on the back of a boat. But it was a brilliant job, and I did it for just over 2 years. And our remit was to map absolutely everything in the North Sea. So I was in charge of identifying everything that lived on the seabed or within the seabed across the entire North Sea, and we just mapped their species distributions, and we did some lovely little CDs of all the little animals that lived there. But at the end of that, I decided, yes, research is for me. I need to get a PhD. So I managed to apply for a PhD up in Napier University in Edinburgh on pollution, which is where the next bit of the talk comes in. But when I throw out that word pollution to you, I want you to shut your eyes. And what is it that you're thinking of? When I say pollution, it's usually very visual, which is where this bit of sugar is going to come in a little bit. But I suspect you might have been thinking, I've got one extra slide here, which I'll just throw in here. It was one of the ironies of our time that while concentrating on the defense of our country against enemies from without, we should be so heedless to those who would destroy it from within. Now, you might be forgiven to think that might be something political from maybe the last year or something. But that was taken from a letter by Rachel Carson, who some of you might know that wrote Silent Spring back in 1953, to the then newly Republican Party in the US. She was particularly concerned about the fact that they were more worried about things outside the country than looking after the habitat within the US. She went on, she was part of the Game and Wildlife Agency, but she was sort of praised with the start of the Environmental Protection Agency in the US, which is currently under threat from the now Republican administration. So pollution. I got you to think about it. Was it something like that? Did you think oil pollution? And these gruesome pictures of these poor turtle and this bird here, absolutely soaked in oil. Or maybe you thought of something like that, lots of dead fish floating on the top of some water pot. Probably some deoxygenation event happened and they all died and floated to the surface. Or perhaps that's what you were thinking. This is quite topical at the moment. Plastic pollution. And these poor gruesome pictures of these wildlife that have swallowed pieces of plastic. What my PhD was on endocrine disrupting chemicals. And the thing with endocrine disrupting chemicals, it's not usually things that you can see. The concentrations that can have effects on the endocrine system of us and of wildlife are absolutely tiny. So the theory behind this, and a little bit of science now, I think you all probably know that you have endocrine glands within your body that produce hormones that get circulated around your body. Once they get to their target cells, they usually attach, this is a cell here, this is the nucleus, to receptors, either on the outside of the cell or within the cell. And your hormones bind to these receptors and then regulate gene expression. Now, what we didn't know up until about the 1990s, well, there's this whole range of pollutants that can also bind to these receptors and either block them from the natural hormones from binding to the receptors, or actually mimic those hormones and produce the same effect that the hormones might have had anyway. So what's worrying about this, because we used to think our hormones were very specific to the receptors to elicit that biological response. What we now know, that lots of these different pollutants can bind to those receptors, but the vast range of different pollutants, oils, plastics, pesticides, pharmaceuticals, surfactants, PCBs, paints, flame retardants, the list could go on and on. The chemical structure of these chemicals varies vastly, but still somehow they can either block the natural hormones from working or stimulate the cells in a way that as if the natural hormone had bound there. One of the first examples of endocrine disruption was noted in around about the 80s, and it was sex changes in the dog whelk here. So it was called imposex, because one sex was imposed on the other. And it was caused by this paint that was put on the undersides of boats to stop biofouling. And this paint contained tributyl tin, or TBT. And at 1 nanogram per litre, it would cause this penis here to develop on a female snail so large that it would block up the oviduct here, which contained eggs, so the animal could not breed, and the populations collapsed. And they collapsed all around the world. And over 200 species this was reported to have been done in. So at 1 nanogram per litre. Now, I suspect most people can't visualize 1 nanogram per litre, which is where the prop comes in today. So I thought I'd do it in terms of sugar and cups of tea. So what I'd like you to do is just rip open your little bits of sugar. Oh, you all jumped at that then. I want you to just sprinkle out about 20 grains of your brown sugar onto the plate. I now want you to separate 2. Okay, a little bit of maths. I'm going to go through it very slowly. It's okay. Right, we're finding effects at 1 nanogram per litre. Now, an Olympic-sized swimming pool holds 2,500,000 litres of water. If we were to get a concentration of 1 nanogram per litre inside that swimming pool, you would have to put in 2,500,000 nanograms of that sugar, or whatever the compound was that we're working with. And 2,500,000 nanograms is equivalent of 250 micrograms, which is equivalent of 2.5 milligrams, which is equivalent of 0.002 grams, 0.025 grams. Now, your average spoonful of sugar is 4.2 grams. The average spoonful of sugar, I've checked this on Wikipedia, it must be true, contains 18,312 grains. That's white sugar. You've got brown sugar today. So if you're going to take 0.0025 grams divided by 4.2, times it by that to work out how many grains that you would need to get that concentration if you sprinkled that in that swimming pool, it's 10.9 grains of white sugar or 2 to 3 grains of brown sugar. So if that was tributyl tin that you had on your plate in front of you, just 2 grains of that sprinkled in an Olympic-sized swimming pool is enough to make a female snail grow a penis. So, and that, 2 little specks of sugar there, I actually weighed on our balance a couple of weeks ago, and it is 2.5 milligrams. It's exactly 2.5 milligrams, in fact. And this is some of the work that we've been doing around Portsmouth here, because our snail population got completely wiped out, as you can imagine, by all the harbours and the amount of shipping that takes place around Portsmouth. We were particularly interested because the snails were only just recovering after they got knocked out in the 1980s. And the red pies here is the proportion of the female snails around our coast of Portsmouth that have got a penis. And the reason why we're quite interested in this, because they've just started redredging all our harbours again to try and fit in our new supersized aircraft carriers. And we're just trying to see whether it completely knocks out our snail population again. So this is an impersex snail here. This is a male. This is the penis here. This is a female. This doesn't have a penis, but this is a female that does have a penis just there. At the same time, there was lots of people reporting intersex fish. These are fish that should either be male or female, but showing characteristics of being both in our rivers, and especially our urbanized rivers and industrial polluted rivers. People are finding, this is testicular tissue, eggs just growing inside the testicular tissue. This flounder, I think, was taken from the tine or the tease. This is its testes that have been dissected out, but this is ovarian tissue that's growing within this poor flounder. And this was believed to be by natural and synthetic estrogens getting into the water. And if you just take a look at this picture here, this is southeast England. Every single little dot on that picture is a sewage treatment plant. So there is a lot. We've got 9,000 wastewater treatment plants in the UK. There's quite a lot in this area, and they empty into our nearest river. The problem is a lot of the pills that we take don't get fully broken down in us. They come out through our urine, they go to a sewage treatment plant, and they don't get fully broken down there either, and then they end up in the nearest river, and then down towards the estuaries. One particular study that wanted to see what were the impacts, the population impacts of having feminized fish, was done by Canadians, and they have the pleasure of having so much land that they can have experimental lakes while we have experimental tanks. And Lake 260 is quite famous because they got permission to dump a load of ethinyl estradiol, which is the synthetic estrogen that you get in the contraceptive pill. And within 3 years, the population of these fathead minnow fish had turned into sex and collapsed. That was at 5 nanograms per litre, which is the equivalent of 10 grains of your sugar in an Olympic-sized swimming pool. Meanwhile, just north of Orlando, Disney World, Lake Apopka, there was a spill of pesticides. This is Professor Louis Juliet. Him and his colleagues, unfortunately he's passed away a couple of years ago, found that there was lots of reproductive abnormalities in alligators because the pesticides were acting and affecting the male and female hormones in the alligators. And similarly, herbicides like atrazine have been shown to cause developmental and sexual abnormalities in frogs as well. Meanwhile, in humans, as a group from California, Anna Soto's group, they've been working on breast cancer development. And there's a particular breast cancer cell line called MCF7, which came from a nun with breast cancer in the 1960s, I think. And it's been immortalized and it's used in labs all around the world. And they usually grow this. And this is quite well known that if you add estrogens to the cell line, they proliferate and grow really fast. They suddenly started working, and their cells started proliferating, and they hadn't even started the experiment yet. And they thought maybe it's something in the plastics. So they rung up the company, because they'd been using the same plastics the whole time, and said, have you changed the recipe for your plastics? And they said, yes. And then they did some digging, and it was actual nonylphenol, which is a surfactant that had leached out of the plastic and caused their breast cancer cell line to proliferate. And then at the same time also, there was reports of lots of developmental abnormalities, sexually developmental abnormalities occurring and increasing in young children. And then in males, there's been a lot of talk about decreased sperm counts and increased sperm abnormalities in us humans as well. Quite controversial, some of this stuff, because not everywhere's finding it. But this all came together where lots of people started thinking, gosh, there's something going on here in the wildlife and maybe us that we should look more carefully at it. And that's why we end up with things on our bottles now, BPA-free on our children's baby bottles, bisphenol A-free. It was found out to be estrogenic. So what we had was a lot of evidence there was things going on in vertebrates, not very much. We didn't know very much what was going on in the invertebrate groups. And these make up 95% of all the animals. Unlike the vertebrates, they had this wealth of knowledge of their endocrine systems and all the things that they could measure from all the biomedical research that went on with these model species that they could then apply to fish that live in our rivers and in our oceans. We didn't have that pleasure when we wanted to go and look at the invertebrates. We didn't have the tools to measure whether they were changing sex, whether their endocrine systems were changing. And that's where my research came in. So crabs are incredibly diverse. The crustaceans are incredibly diverse. I had to choose a particular species that, one, didn't change sex naturally, because there are lots which can change from males to females and others that change from females to males naturally. They're hermaphrodites as part of their life. There's some like this one, like this Daphne, that can clone itself over and over again. It doesn't use sexual reproduction. So the animal I chose was this one, and it's called Echinogamerus Marinus. It was named about 200 years ago as Gamerus marinus, but it's changed its name a lot since then. And this you'll find on any rocky shore around our coastline, and it reaches right the way down from Portugal, right the way up. It used to be on the southern tip of Iceland, but now with the changing climate, you can find it right the way around Iceland. we started looking at clean and polluted sites. I was based in Scotland at the time, and thankfully SEPA, the Scottish Environment Protection Agency, had classified all their bays and beaches and estuaries based on whether they were highly polluted or clean. So we started looking at a number of different clean and polluted sites. And this is one site which is particularly bad, and it was called Inverkeithing Bay, which is within the Firth of Forth. And there was a paper mill here and an old scrapyard, a breaker's yard here, and this is one of the sister ships to the Titanic coming in to be broken up there. It is stinking, this particular bay. It's full of all sorts of pollutants, metals, PCBs, some of the wastewater coming out of the paper mill. And what we found was about 30% of the amphipods, these little crustaceans I was talking about, were intersex. They were not quite male, not quite female. Whereas in the clean sites we were looking at, it was only 1 to 3% of the animals. So we thought, aha, we found something as well. We found pollution causes sex changes in these crustaceans. We found different morphotypes, phenotypes of this intersexuality in these crustaceans as well. These we called normal females, and these we called normal males. And then we had some intersex females and then intersex males. And superficially, they look very much the same. I'm sure you can't tell the difference between them. But when you look on the underside, as we'll do on this next slide, these are genital papillae. These are the male bits that you should find on one of these little amphipods. This male over here has got its little genital papillae here, but it's developing these little appendages coming out from the bottom of its legs. These are brood plates. These are appendages that you should only find on a female to keep the eggs in place. This is a normal female. These are these brood plates with lots of interlocking hairs that keep the eggs in place. This is a female over here, but this one has developed genital papillae that you should only find on a male. We started looking at these animals in a little bit more detail, and what we found was they were full of parasites. Not one, but two particular parasites. There's one called a microsporidian. These are these ones here. And we recently found that whenever we found one of these microsporidian parasites, we also found a Paramyxian parasite as well. And these are very clever parasites because they can feminize their hosts. And when we look at the proportion of the animals that were infected and weren't infected, we found that our normal males and our normal females weren't that infected. Whereas our intersex males and our intersex females, in red, this is the proportion that was likely to be infected. A large proportion were them infected. So we thought, okay, we've solved this. This is probably a parasite issue, and it's the parasites that are causing the intersex in these animals. When we started dissecting some of these males, though, we found that internally, they had characteristics of being male and female as well. So the story wasn't as simple as we thought. There was some of these, despite not very many of them being parasitized, were showing signs of intersexuality as well. This is what the parasite does. It's very clever. It gets passed on from female to female. But if it ends up in a male, it's a dead end for the parasite because it's only passed on via the eggs. And these animals typically produce about 40 eggs, and it's usually a 50-50 sex ratio. 50% females, 50% males. But if the parasite ends up in any of the males, it can feminize them, stop them from turning into a male, and they turn into a female. Sometimes it doesn't fully complete that process, and they get halfway and turn into an intersex specimen. What we found internally, this is a male here, this is its testes here. We found female oviducts developing on the insides of them, and we found vas deferens, which is a male structure, developing on the backs of the ovaries of the intersex females. As a result of these abnormalities, we found that the males produce less sperm. We found that the intersex females produce less eggs. One of the things that the people studying the fish and the intersex fish and the feminized fish were having difficulties with was what could the population level effect be? Because fish produce tens of thousands of eggs, you can't go counting them to see what proportion might be might be impacted by feminization. We're lucky because they only produce about 40 eggs. We can count the sperm and we can count the number of eggs fertilized in intersex specimens. And as a result of that, I was able to team up with Dr. Irene Martins in Portugal over 10 years ago now, and we produced that, which I'm not even going to attempt to explain to you other than it's a population model to try and work out what the implications are of intersexuality in the population. In a very simplistic way, you have juveniles, some of them turn into males, some of them turn into females, and then some of them turn into intersex females. Depending on the temperature, the salinity, and everything that's going on in the environment, it affects their mortality, their growth, the number of eggs they produce. And we can fiddle around with the number of insects in the population and model it over 10 years, and we worked out that at certain levels of intersexuality, they would crash within about six years. So we could do some predictive work about what the implications of pollution feminizing our wildlife could be. So I'm taking it from the individual level right the way up to the population level effects of these animals changing sex. Next thing I wanted to do is take it down to the molecular level. So we had this question of whether pollution was increasing parasites, which then caused intersexuality, or was pollution directly causing intersexuality or the demasculinization of our animals. We needed some sensitive molecular tools to measure these sex changes, like the fish people had. Now, the cost of the human genome was 2.7 billion, and it cost about 15 years. Now, a genome costs about $1,400, and you can see the price has come down substantially. We're fortunate enough to get a substantial grant from the Natural Environment Research Council, and I teamed up with a great molecular biologist, Professor Pete Keel in Cardiff. And what we did, we wanted to produce a genetic blueprint of the genome of our animal and all the genes that could be transcribed from that genome as well. And during the time that we did this, the technology kept changing quicker than we were actually doing the science. We didn't just want to do one, we wanted to do it for our normal males, our males that were intersex, our females, and our females that were intersex, and whether they had parasites or not. And we did those for 4 different tissues inside the animals as well. And this was done by some of our postdocs and some of our PhD students. And what we ended up with several billion sequences that took computers several weeks to scrunch, and then we end up what you could roughly call 43,000 genes for these animals. And then from that we could work out what genes were more likely to be expressed in a male and what genes were more likely to be expressed in a female. And there was 2,500 and around about 5,000 female genes. From that, we wanted to know whether any of our males were producing any female genes. Were there more of these genes in an intersex male compared to a normal male? And I'm just going to show you one example of this, because I know this is quite heavy on the molecular science. We just found this one gene. It's called protein TIS11. These are our females along the top here. This is a normal female. This is an infected normal female. This is an intersex female. This is a normal male, an internal intersex male. On the outsides, it looked fine. And this is an external intersex male that was infected. And you can look at the number of times we've got a sequence for that particular gene. It's up in the tens of thousands for the females. So it's a very female-expressed gene, but very low in a male, but getting increasingly bigger as the animal got more female or less male. So then this might be a good marker then. We could go to a beach that's polluted. Is this gene being expressed in a male? This is a very subtle indicator that it might be changing sex. When we looked at this gene in other animals as well, this is a fruit fly. It was highly expressed in an ovary and wasn't expressed very much at all in a testes. That gives us another clue. This was a really, really good gene to be looking at. And we came up with hundreds of these, and we've been looking at them in lots of different beaches. So we had a molecular toolkit that we could go right the way around the UK and actually measure clean and polluted sites. And that's what we've been doing and in the process of publishing now. One of the things that we found out was that sex determination in lots of vertebrate species is like a seesaw. you've got female on one side, male on the other. And if you make something more female, if you tip the seesaw this way upwards, they get less male. Similarly, if you make something less male, it becomes more female. And that kind of happened roughly across all the vertebrate species. What we found in our animals was if you make them more female, whether it be through pollution or parasites, they don't get less male. If anything, they get a little bit more male. They upregulate all their male genes. So they could be said to fighting their inner sex change. And it makes sense, really, because if you've evolved with a parasite that can change your sex, and if it completely changed all the males to females, the population would crash. So it makes sense to have a biology that could resist changing sex. So I'm going to leave the sex-changing stuff now, and I'm going to go on to the brain-bending chemicals now, the Prozac, and this is something that has got a lot of attention from the public that we've been doing for obvious reasons. Now, as I mentioned earlier, we take lots of pills. They don't get fully broken down. They end up in a sewage treatment plant, or if you, like me, you've got kitchen drawers full of lots of headache tablets and things, and they go out of date, and then you chuck them in the bin, and then they go to landfill, and then they leach out, and then they end up in the nearest river anyway. Or what we should be doing is take them back to the pharmacy, and they should incinerate them or something like that. We don't do that very much in this country. I think the take back in this country is about 10%, whereas in other countries in Europe it's considerably higher. But effluent water coming out of a sewage treatment plant may contain over 1,000 different pharmaceuticals. But the one pharmaceutical I'm interested in is the antidepressants. Now, if you look at the United States, over 1 in 10 people take antidepressants. The UK is somewhere here, 71 out of 1,000, but that varies considerably throughout the UK. Some parts of the UK, it could be 1 in 5 people taking antidepressants. That's a lot. 1 in 10 is about the equivalent of the same people taking the contraceptive pill, taking into account that the contraceptive pill is only taken by one sex and within a small window of their reproductive life. And this is how they work. This is a nerve here. It's sending down the signal down to this next nerve here, and what's happening, this serotonin is released into the synapse here. But what a lot of the most common antidepressants do, the selective serotonin reuptake inhibitors, they prevent serotonin from being taken back up into the nerve. So it accumulates and gets more concentrated in the gaps between the nerves. And this is not strictly true, but it makes a nice little thing going on here. And the fact that it just demonstrates that we have these neurotransmitters on our brains, dopamine, serotonin, oxytocin, that control our behaviors. But these things, serotonin, dopamine, and epinephrine, control lots of other different physiologies within wildlife as well. Reproduction, growth, maturation, metamorphosis, metabolism, immunity, as well as behavior. But it's behavior that gets most amount of attention because the drugs like antidepressants were designed to change people's behavior. But serotonin can be found, it has all these functions across all these wildlife. Now, the first person to have done a study like this where they exposed an animal to Prozac got the Ig Nobel Prize. And that is a kind of fun version of the Nobel Prize Awards that's given out to science that makes you first laugh and then makes you think. And what they did, Pete Fong exposed these little zebra mussels here to Prozac because he knew if you put serotonin in the water, you could get those little bivalves there to spawn. And if you're breeding these, if you want the males and females to release their eggs and sperm at the same time, if you chuck a chemical in the water, it would make aquaculture a lot easier. So he thought, well, let's just chuck Prozac in the water and see whether it does the same thing. Prozac is designed to increase serotonin. And it did. And some within minutes, 70% of the males within an hour had started spawning. And this is in molar concentration here, but it was around about 300 nanograms. You can do the maths with the sugar if you want to for that one. But so it's a little bit more. Obviously, it's probably a couple of 100 grains of sugar in an Olympic-sized swimming pool in terms of the concentration of Prozac to get this to spawn. Now, what are the concentrations in our environment? Well, they can be up to 1 microgram per litre. That's 1,000 nanograms. But they're mainly found in this concentration, around about 20 nanograms per litre. So that's about 2 times that, which is about 40 grains of brown sugar in an Olympic-sized swimming pool. That is the concentration, if that was the weight of the actual Prozac, put into an Olympic-sized swimming pool and given a stir. I got particularly interested in this because I was aware of parasites that could alter the serotonin in the brains of my animal. This in yellow here is serotonin lit up in the brains of these little amphipods. And it's done by these parasites here. They deliberately upregulate serotonin in the brains of these animals to change their behavior to make them attracted to light. And they do that for a very good reason, because unless this parasite here gets inside a bird or a fish, it can't change its life, it can't complete its life cycle. So these little animals usually stay in the dark underneath seaweed and stones, but if they've got a parasite, they're more likely to swim out into the open, and as a result of that, they're more likely to be eaten. So we thought, well, if a parasite altering its serotonin can change its behavior, what happens if we put a little bit of antidepressant in the water? Can that change its behavior and a similar behavior as well? So this is the kind of experiments we did. We got some of these animals and we put them in a very simple tube, and it literally was this tube. And we scored every 30 seconds how far up the tube it was with animals that didn't or did have parasites. And the ones that did have parasites were found a lot higher up this tube than the ones down here. And then we had another bit of tubing that had a black bit of tape. This was how simple this experiment was. And every 30 seconds, we'd score it 0 or 1, whether it was on the light side of the tube or on the dark side of the tube. And again, whether it had parasites or it didn't have parasites. And if it had parasites, you get a really big score that indicated it spent the whole time on this side of the tube. And if it didn't have parasites, it spent the whole time on this side of the tube. So this really neat behavioral experiment for testing whether antidepressants might also change this behavior. We also threw in the serotonin in the water, and the more serotonin that we put in, the more time they spent higher swimming up the water column, and the more time they spent in the light side of their tubes as well. So that was just pure serotonin. What we hadn't done yet was put Prozac in, which is called fluoxetine. What we were wondering was, if we did alter their behavior, would there be a knock-on effect on the population? You're 28 times more likely to find an infected one of these animals inside the stomachs of these fish than an uninfected one. So by changing that behavior subtly, it is substantially more likely to be eaten by a fish or a bird than one that's not infected. And this is what we did. We put Prozac in the water, and it increased the amount of time it spent on the light side of these tubes, and it increased how high they were swimming in the water column. And this was done at around about 20 grains of sugar. It was about 10 nanograms and 100 nanograms were having a significant effect. We got a bit more sophisticated with our experiments, and we bought this machine called DanioVision that's got a fancy bit of software that can measure the behavior of our animals swimming around in their little dishes. And it can measure their behaviors 100 times per second, and it can do about 20 different behaviors. It can tell you how fast they're swimming, how many times they change directions, the angle at which they change direction, how much time they're active or not. And you get lovely little traces like this, which you can do some statistics on. And this is what we did, and we published this result. We'd actually driven down the effect that we could detect, the effect of Prozac in the water, down to just 2 grains of sugar, which was just 10 nanograms, or 1 nanogram I think we even got down to in the end. It was just the black line of the controls. We switched the light on to shock them. They settled back down. We switched the light on to shock them. Switched the light on to shock them. Green behind is sertraline, which is one brand of antidepressant. The red one behind that is fluoxetine, which is Prozac. As you can see, they're a lot more active. They're swimming a lot faster in the ones that we exposed to very low levels of antidepressants than the ones that we didn't. And we've done that with lots of different antidepressants now, and we're finding some really interesting results. Chanel is somewhere in the audience. She's doing this as part of her PhD now. If you watch this little shrimp swim around here, you've got the dark side and the light side, and in a minute, the light's going to come on. Light's on, it's going to recognize the light's on, and it's going to shoot over here and go a little bit mad. I think he darts back into the light side a bit and realizes, oh, I don't want to be over there again in a minute. Oh, yeah, didn't like being over there. So we can now measure quite complex behaviors. I think the computer's starting to go a little bit slow, because in a minute the light's going to go off and he's going to be quite happy to go on that side again in a minute. Yep, he's quite happy over there now. So what we're trying to do now is see if we can measure anxiety in some of these animals. Not just phototaxis, how attracted they are to light, is whether they reduce their anxiety on some of these medications that we find detected in our environment. One of the other things we want to do, because we produce this genetic blueprint, this atlas of all the genes that they have inside these animals, we wanted to know what the gene expression was going on in these animals once we expose them to Prozac. And again, we haven't published this results yet, but we're getting some really, really interesting data. So this is the genes or the gene pathways that are being changed in the heads of our shrimp at an environmental level of antidepressant. Literally about 20 grains of sugar in an Olympic-sized swimming pool. And some of the things that are really quite shocking us, but we really need to back them up with some more science, is the fact that we've got Alzheimer's disease, Huntington's disease, and there's something, it's a Parkinson's disease coming up. We've got lots of different neurotransmitters being affected, and we don't know, is our shrimps being affected? Are the nervous systems being affected to the extent where they're getting neurological disorders? Is wildlife being affected in this way? And we simply don't know, and this is what we're hoping to take a lot further now, because we find that result very intriguing. Other people have done some fascinating stuff with some antidepressants since we started doing our work about 7 years ago. This is a group in France that found out that, as most of you know, cuttlefish are really intelligent organisms. And there's a load of shrimp in this glass jar here, which they can't get at. And the control animals will strike at this glass jar a couple of times and then work out they're too difficult and swim away. But the animals on just 10 and 100 nanograms of Prozac, fluoxetine, carry on striking. They don't learn. They carry on striking at those shrimp. Not only that, you can take those cuttlefish away, bring them back several weeks. The control animals had learnt, and they remembered also that it was difficult. The ones on fluoxetine hadn't learnt, and they carried on striking away at that. And that was on environmental levels of Prozac in the water. Now, the pharmaceutical industry are making use of the fact that you can look, there's pretty well-defined behaviours of fish when designing their anti-anxiety medication. If any of you have kept a fish tank before and you've gone to the fish shop, got a new fish, put it in your fish tank, it will go and hide in the corner somewhere for a little bit until it feels brave enough to come out into the open. And there's a novel tank test that you can do where you get a fish, you treat it with something, and you have controls, you put it in a tank, and you can look at the different behaviours. And this is the behaviour of something on ethanol, on morphine, on caffeine, alarm pheromone. That's if it can smell another fish. It dives right the way down to the bottom. And then you've got the effect of chronic fluoxetine. In a fantastic study that was done in Sweden, and I might need to press start on this, they got permission to put fish that they'd exposed to anti-anxiety medication in a lake that didn't contain fish. It was right in the far north of Sweden. And what they did, they exposed the fish to these anti-anxiety meds. they put acoustic pingers inside the stomachs of the fish and then acoustic transponders in a big grid across the lake so they could triangulate where any of the fish were at any one time to replicate what's going on a tank. Do the fish that are on anti-anxiety meds go into the center a lot quicker than the control fish? So I'm just going to press play on this. And those are individual fish. The red ones are the ones exposed, the green ones are the controls, and the blue ones are pike that they threw in there for good measure because they wanted to look at the predator-prey dynamics of it. And essentially what they found is the ones on the anxiety meds were more likely to swim in the center of the lake than the ones that are not. But it showed that the things that we're finding in the labs could be extrapolated to the field as well. Now, when we did our Prozac work, we've got a fantastic press office here, and we put out a press release, and it went pretty berserk. This is an article that they did at National Geographic. Prozac is making shrimp reckless. It got 2 million tweets. And then there's another one, NPR, the radio station in the US, Science Direct, I can't say, Telegraph, 800,000 tweets, 450,000 tweets, 350,000 tweets. Enormous reach. And this went quite big, and I spent a lot of time doing lots of radio interviews. And what you find and work out as a result of that is if you've taken up all the radio listenerships, the readerships of all the newspapers, and all this reach that's gone through social media, you've reached 1 billion people. You've reached 1/7 of the planet. And that's quite extraordinary. And then the implications of that, and the impact is then suddenly you have politicians that are asking questions on your behalf in the European Parliament, and not just one political party, numerous political parties, and insurance industry doing big double-page spreads on the science, because then they're getting worried about these new pollutants. Because if you think about it, we are a growing, aging, increasingly medicated population that is becoming increasingly dependent on clean water and in a changing climate. So there are implications for society as a whole as well. Artists started getting interested in their work, and there were some brilliant cartoons done in the newspapers. And there's even a play that started touring in March 2017 called Shrimp Dance, where it's an alternative interpretive dance, and it's all based on this science, which is really fantastic to see the science getting taken into the arts arena. So then that brings me on, I've just got 5 minutes, to the last bit of my talk, science integrity and science communication, alternative facts and fake news. And it's British Science Week. Is science broken? I thought I'd ask that brave question. This is an article literally just 2 weeks ago. A crisis of trust is looming between scientists and society. It's time to talk. And I'm sure most of you know about the problems with vaccination of MMR, and the fact that there wasn't a big take-up rate, and then measles and mumps and rubella are on the increase. And we're losing the battle with that in some countries. In other countries, there's still lots of people worried about going for the MMR vaccination, and other people get saturated with newspapers saying that they've found a cure for cancer, and they're losing trust to a certain extent in scientists, and there's something that we need to do better. Now, my own research was getting misinterpreted. If any of you know Alex Jones, he's a well-known conspiracist, friend of Donald Trump in the US, and he's there saying that, yes, there's people deliberately putting this medication in the water, and he goes on to talk about massacres in schools and various different things like that with people on antidepressants. Pollutants in water making shrimp suicidal, putting babies at risk. And I don't think our shrimp study ever mentioned anything about babies, but there's other people, because it's quite controversial, because they're using this suicide term. There's lots of people that are thought to commit suicide after being put on antidepressants rather than to prevent them from harming themselves. So it's really tapped into their political agendas, some of our research. And we have a problem. This expensive global warming bullshit has got to stop. Our planet is freezing, record low temperatures, and our global warming scientists are stuck in ice. I think people in this country have had enough of experts. Are we losing this battle? Is science broken? Is there something we could do better? Probably yes. Now, do scientists need to get their own house in order? And this was a really interesting study that I'm going to show you next. And it's the use of positive and negative words in scientific abstracts between 1974 and it's published in the British Medical Journal. And I can't see that. Amazing, astonishing, novel, groundbreaking. We're using positive superlatives much more in our science. We're trying to egg it on more. And some people believe that that's because of increasing pressure to publish and gain research income in an environment where it's harder to do so. So is this a result of being in a publish and perish culture? Are we exaggerating our work more? Is that exaggerations being picked up by the press more? And is that some of the discontent that is amongst the general public? Bigly as well might be a new one that might come in soon. And I've done this for the environmental journals as well. That was done on medical journals. So I got the top 10 environmental toxicology journals, and I used all the same terms that they did. And we're finding exactly the same. A substantial increase in the positive superlatives used in papers, whereas negative ones are pretty much flatlined, despite the fact there's been a huge increase in publications across that. And us, in the area of environmental toxicology, we have this problem where we can't, if we don't find anything, we can't really get it published. So all our negative results go in the bin when they should be published, so you can compare against the positive results. So where they're all mouthing off about these things having a toxic effect, we've supposedly got this axe hanging over our head that if we don't publish and things like this, then we might not be able to get a job. So is that a problem for our institutions as a whole? Do we need to look at this? And I just leave you with one example. The Ban the Beads campaign has been hugely successful. Quite rightly, quite ridiculous that we should have little plastic particles in our cosmetics and in our toothpaste and things. But the extent to which us scientists are really pushing this, and to be fair, you expose animals to these and you do find them in their guts and they do transfer their guts, and if you feed different animals, those animals, then they can pass along as well. But maybe we're not getting things in context enough. That is the amount of plastic waste that goes into our oceans every 15 seconds. So is that really the problem where a lot of our emphasis is at the moment? Yes, it's great, it's banned, completely non-necessary. But really, is that what we should be concentrating on? It's slightly ironic that I've given you all plastic plates for today. But on that, I'd just like to thank you for listening.